Hello, beautiful biology students. We are going to zip through Chapter 13, Lessons 1 through 3 in preparation for our unit test. We'll begin our discovery of DNA with Frederick Griffith's experiment. Remember, in this experiment, he wanted to figure out how it was that certain bacteria were killing mice. In the first Petri dish here, he has these pathogenic S-type bacteria that actually end up killing the mouse. Then he has another Petri dish containing harmless R-type bacteria where the mice live. He then heat kills the S-type bacteria that's harmful, and lo and behold, the mouse lives. Now let's look at the experimental group. In the experimental group, he has the harmful S-type bacteria. He has the harmless R-type bacteria that he is putting inside the mouse. And suddenly the mouse dies. So what Griffith concludes is that something, he's not quite sure what, has moved from the S-type to the R-type bacteria, transforming the R-type into the harmful S-type, which then killed the mouse. Now Oswald Avery will come along with his team of scientists, and they want to figure out exactly what is it that was transferred from the S-type bacteria to the R-type bacteria to kill that mouse. He'll discover, after testing many different macromolecules to include carbohydrates, lipids, ribonucleic acid, as well as deoxyribonucleic acid and proteins, that indeed it's DNA that is the genetic material. This is a really, really important discovery because up until then, most scientists thought proteins were the genetic material. Oddly enough, scientists still don't believe that DNA is the genetic material. So along come two scientists, Hershey and Chase, and they are going to work with viruses to prove that DNA is in fact the genetic material. Specifically, they are going to work with bacteriophages. Now, these are a very specific type of virus that only infect and kill bacteria. But like all viruses, they have a protein coat and they have genetic material. In this case, it's DNA, but sometimes it can be RNA. Now, in today's world, we now know that viruses have two different cycles. They have what's called the lysogenic cycle, in which case the virus is injecting its genetic material into the bacterium, and you can see it right here. And the bacterium is actually incorporating the viral genetic material into its own ring of DNA. And then every time the bacteria divides, it's going to carry that viral information in its daughter cells. And in the lytic cycle, the virus here is injecting its genetic material into the bacterial host and actually turning the bacterial cell into a viral factory until those viruses hatch out, destroying the bacterial host, and they'll look for a new host to invade. So what Hershey and Chase want to find out is, what is that exactly that the virus is injecting into the bacterial host cell that obviously is going to get replicated inside each daughter cell and then tell that bacterial cell or give it instructions to actually make new viruses. So Hershey and Chase are going to use two radioactive labels. They're going to use phosphorus and sulfur. And the reason why they're choosing these two elements is because nucleic acids unlike other macromolecules, contain phosphorus, and proteins, unlike the other macromolecules, can contain sulfur. Now remember that all viruses contain genetic material inside the head or protein coat or capsule. And then, of course, the capsule itself is coated with proteins. So here we can establish our independent variable as the radioactive label being used, whether it's phosphorus or sulfur. And then over here at the far end, we can see that the dependent variable is whether or not there is radioactivity inside the bacterium. And lo and behold, there's only radioactivity inside the bacterial cell in which they labeled phosphorus, which proves that, in fact, DNA is the genetic material. So we'll wrap up this lesson here. We now know that DNA has three major roles. One, it is a storage site. It stores information for making proteins. Two, DNA has to be replicated. It has to be able to copy itself every time a cell is going to divide. And then finally, when proteins are needed inside a cell, genes in that DNA molecule must be expressed in order to make the proteins it codes for. We'll move on now to Chapter 13, Lesson 2 on DNA structure. Now, when you look at DNA, say on Google, and you Google images, you may find lots of different renditions of DNA. Sometimes it looks like this, sort of two-dimensional, and then other times you see it really complicated, and this might look a little bit scary here, but this is showing the atomic structure of an actual DNA molecule. We'll be sticking to rather simple renditions of DNA so that we can learn all the parts. So let's take a moment here and just review exactly what we mean when we talk about DNA. So DNA is raw nucleic acid, and that is shown here in the diagram. Again, it looks like a twisted ladder. But in order to fit it into this itty-bitty living space that we call the nucleus of a eukaryotic cell, we need some proteins to get involved. And you can see that in the image here, where we have DNA being wrapped around proteins called histones to form nucleosomes or beads on a string. And then, of course, this further wraps around more proteins until you get this full-fledged chromosome. So for the rest of this lesson, we will just be sticking to the DNA level, and we are going to not really be talking about the proteins that are involved in wrapping it up. So remember that DNA is a polymer, deoxyribonucleic acid, and all polymers are made up of repeating subunits or monomers. And in this case, the monomer of DNA is called a nucleotide. So what is a nucleotide made up of? Nucleotides are made up of three parts: a nitrogenous base, a 5-carbon sugar called deoxyribose, and a phosphate group. And if you look at the image right here, you can see that that phosphate group has a slight negative charge, which gives the DNA molecule a slight negative charge all around. The nitrogenous bases are categorized by their size. There are two groups, the pyrimidines, which are composed of a single ring, so they're going to be smaller, and they include the bases thymine and cytosine. The purines, they are bigger because they're composed of double rings, and they include adenine and guanine. And if you remember in class, we talked about memorizing this as pure ag. So the purines include adenine and guanine. Although DNA looks like a ladder, it's actually anti-parallel. meaning that one side of the DNA molecule will end in a sugar, while the opposite side ends in a phosphate group. So we simply label the sugar end as a 3', and we label the phosphate group end as a 5'. You can see that over here as well, phosphate group 5' and sugar 3'. Notice also that we have base pairs that are labeled. The bases actually make up the rungs of the DNA ladder, and they are held together by hydrogen bonds. Let's see if we can label some of the parts of our DNA molecule. First of all, we have a sugar, shown here with the black pentagons. The white rods represent the phosphate groups. The colored rods represent the nitrogenous bases. You'll notice that the bigger rods represent adenine and guanine, and the smaller rods represent the pyrimidines, which include cytosine and thymine. In between our base pairs, we'll have hydrogen bonds. We'll also have a bond between each nucleotide on the sides of the DNA ladder, and these are called our phosphodiester bonds. So again, just to review here, the sides of the DNA ladder are made up of phosphates and sugars, called the sugar-phosphate backbone, and the rungs of the ladder are composed of base pairs, two bases held together by hydrogen bonds. Now, a great way for you to practice naming all the parts of a DNA molecule is just using your own two hands as shown in the photo. You can see that her hand represents a DNA nucleotide. We have the phosphate shown with her index finger. Her palm represents the deoxyribose sugar, and her thumb represents the base. Right here would be the hydrogen bond to the next base on the adjacent nucleotide. Here's her sugar, and here's her phosphate group. Using Chargaff's rules, if this base here is adenine, then we know that this base here is thymine. And if this base here is guanine, then we know that the base here is cytosine. Notice that whenever you have a purine on one side of the DNA ladder, you'll have a pyrimidine paired with it. So let's do some practice here. We have a matching exercise. Let's start with number 1. Number 1 is pointing to two bases or a base pair. Number 2 is pointing to a phosphate group, a sugar, and a base. This is known as a nucleotide. Number 3 is pointing to a single ring base. This means it has to be either cytosine or thymine. Using Chargaff's rule, we know that this would have to be thymine, and thymine is a type of pyrimidine. Number 4. Number 4 is pointing to this sugar called deoxyribose in the DNA molecule. Number 5 is pointing to a phosphate group on the sugar-phosphate backbone. And number 6 is pointing to the dashed lines holding the two bases together. And this, in fact, would be our hydrogen bond. You can see that some bases will be held by 3 hydrogen bonds. That's between cytosine and guanine. And adenine and thymine are only held by 2 hydrogen bonds. Now let's look at number 7. Number 7 is pointing to a bond that's holding one nucleotide to the next nucleotide. This is called our phosphodiester bond. We can shade the phosphodiester bonds now between the nucleotides like this on one side of the DNA ladder and like this on the other side of the DNA ladder. To wrap up lesson 2, let's talk about the scientists involved. So way back in the 1940s, we have Rosalind Franklin working under Maurice Wilkins, and she is going to discover that the DNA molecule has this twisted ladder shape. She will coin the term double helix to describe that shape. You can see in the photo here, she used X-ray crystallography, and you have to remember it's a bird's eye view of the DNA molecule. And finally, we have Watson and Crick, who used Chargaff's rule as well as the work of Rosalind Franklin to build this really large molecular model of DNA. So they are attributed with actually discovering the molecular makeup of the DNA molecule. And that concludes our review of Chapter 13, Lessons 1 and 2.